Hey everybody, in this video I am going to describe lentiviral vectors. Now these are things that can be used to insert genes into the genomes of certain cell lines, and they can even be used, and they are being used in gene therapy approaches for certain human diseases. So let me start with some definitions. So a vector is essentially, what can we say? So any vehicle, I'm going to put vehicle in quotes there, used to insert DNA into a cell. So different types of vectors we could use in molecular biology might be virus-based vectors, which I'm going to talk about today, and even plasmids. Plasmids can be considered vectors. So now, okay, we defined vector. Now, what is a lentivirus? So we've covered lentiviruses in the last couple of videos. We covered one, I should say, in particular. So HIV-1. So HIV-1 is a lentivirus. HIV-2 is a lentivirus. And I think these are the only two known lentiviruses in humans. So there are other lentiviruses in other primates, in other mammals. I think feline immunodeficiency virus is a lentivirus. So there are lots of different lentiviruses. As far as I know, I think HIV-1 and HIV-2 are currently the only two known lentiviruses in humans. I could be wrong on that. It's not important to memorize that for the quiz. So another term which might come up is transduction. So we use the term transduction when, say, researchers are using or trying to insert foreign DNA into a cell with a virus. So, and also transduction occurs naturally, right? So there are, when viruses insert their DNA into the genome of a host, they are transducing the host. So transduction, that's what that word means. And we can use transduction in molecular biology when we use a lentiviral vector to introduce DNA into a host cell. So we are going to focus on a specific class of lentiviral vectors called third generation lentiviral vectors, and I may abbreviate lentiviral vector as LVV from time to time. And let me use this piece of paper right here with these notes already diagrammed on here. So in the last couple of videos, we talked a lot about HIV-1 and the structure of HIV-1. So essentially, the lentiviral vector is going to be very similar to HIV-1. It's going to have an envelope. It's going to have envelope proteins, which are going to allow the viral vector to fuse with the host cell. In this case, it's GP120 and GP41, the HIV proteins. So this viral vector right here, if this isn't HIV-1, if this is an example of a viral vector, it's going to be really good at infecting or transducing T cells that have the CD4 receptor, because remember, this is GP120, GP41, they mediate infection of T cells based on the interaction with the CD4 receptor. So the viral vector is going to need the matrix protein, it's going to need the nucleocapsid protein that is around the, binds to that DNA that we need to get into, or RNA, right? RNA that we need to get into the cell that will be turned into DNA. by the reverse transcriptase. So reverse transcriptase will need to be there. Integrase will need to be there to get that into the genome. The protease will need to be there to form this structure. The capsid will need to be there. So all these things, all these structural proteins that we talked about when we talked about HIV-1 biology, all of these pieces will need to be inside the lentiviral vector. And what we could do, let's say we wanted to, say we had a cell line here. We'll start simple before we talk about gene therapy. We'll just talk about transducing cell lines for research. Let's say we wanted to introduce a protein or a gene into these cell lines to study the gene. And we'll take a simple one. The gene we will use will be green fluorescent protein. So for fun, let's assume that we have a cell line, say it's a mammalian cell line, and we want to make these cells glow under UV light. So we want to have them express green fluorescent protein. So we want to put a gene for green fluorescent protein into the genomes of these cells. Well, we could use this virus right here to do it if the GFP gene is in here, inside this little RNA right here. If this RNA molecule has the coding sequences and the promoter elements and everything needed to express GFP in these cells. So what I'm going to do is cover how we can make these lentiviral vectors. So this is essentially a virus or virion. But it's not a natural virus, right? We put this together with the specific intention of putting a transgene into the genomes of these cell lines. And I'm going to explain how we put these together. That's why, isn't this a virus? How come we keep calling it a lentiviral vector? Well, technically, yeah, it's very similar to HIV. It just doesn't have the HIV genome in here. So I'm going to refer to this as an LVV, lentiviral vector. So to make a lentiviral vector, what are we going to do? Well, we need a packaging cell. So we're going to need a cell. We're going to need cells, so mammalian cells, because these are mammalian lentiviral vectors. We're going to need a mammalian cell to build these little viruses, these little LVVs for us. And a very common one that is used is called HEK293. So this is a cell line, human embryonic kidney cells, number 293, initially isolated in 1973, and now they're used by labs around the world that study mammalian biology. So it's a cell line, very common cell line for researchers who study mammalian biology. So first step is to transfect. This is another term which might be new to most of you. So transfect is a term mammalian biologists, mammalian molecular biologists use when they're introducing DNA or RNA into a cell line. So they're transfecting the cells with DNA. So we need to transfect these cells with 4 different plasmids. And the 4 plasmids make up the 3rd generation lentiviral vector system. I'll describe these in a moment. So transfect with these cells, with these 4 plasmids, and some of these cells become LVV, lentiviral vector producing cells. In order for that to happen, a cell would need to take up each of the plasmids. And that process is very inefficient. And if you go through the notes, you'll see one of the major hurdles to reducing the cost with LVV gene therapies is making this process more efficient. Because if only a few cells in this population of cells take up all 4 plasmids, only a few of those cells are going to produce the LVVs. So after this, after you have some LVV producing cells, LVV producing cells, I should say, not LVV producing vectors. After you have the cells producing the virus, okay, the virus is going to bud, the viruses will bud from, the LVVs will bud from the cells, and they can be collected. And they can be preserved. We can say cryopreservation. So that means you can store them at, say, minus 80 degrees Celsius, so a really cold temperature. And anytime, so once you have these LVVs, anytime you have some cells that you want to make green, if we want to make all these cells green, what we need to do is we just need to mix our LVV with these cells. I'll put these little circles here. What these are the little LVVs, and they're fusing with these cells. They're going to insert that transgene carried by the LVV into the genomes of the cells. So most of what we're going to talk about is, what are these 4 plasmids? Why do we need 4 plasmids? Well, one of the reasons we want 4 plasmids is, all this was derived from HIV-1, right? And so researchers, what they want to do for safety reasons is they want to split all the necessary parts of HIV-1 across 4 different plasmids to, keep the process from, causing an HIV-1 infection. So if you split the different components of HIV-1 that are necessary to make the lentivirus vector, if you spread them across 4 plasmids, it's very unlikely that this will actually create a virus which is going to be harmful to humans. So I just had a sip of coffee. I wonder how loud my swallowing was there. Okay, so what are the 4 plasmids? So different researchers and different companies that make these systems call them different things. So one of them is called the packaging plasmid. Now, I'm going to go over what we will refer to them as: a packaging plasmid, an envelope plasmid, a regulatory plasmid, and a transfer plasmid. So let's take the packaging plasmid first. And if I forgot to mention, you really need to make sure you watch the HIV-1 videos before you watch this, because I'm assuming you watched those videos and understand HIV-1 biology and organization of the genome. So I won't be covering all the little details. I'm going to assume you know some things. So it may get really complicated really fast if you didn't watch those videos yet. So let's say here's our packaging plasmid. So this is one of the 4 plasmids that is involved in or needed for the LVV system. So now plasmid is a circular molecule, and let's say the important part that's going to encode the proteins needed for the viral vector is here. I'm going to diagram that down here. So there's going to be a promoter, and what the promoter is for expression of the genes on the plasmid in the packaging cell. And there'll be a +1 site for the transcriptional start site. And then what we're going to have here is the GAG gene from HIV-1. And remember, and I have to diagram it in this weird way, right? Because the POL gene slightly overlaps with GAG. So we're going to have the GAG gene and the POL gene here, or the coding sequences, I should say, for the GAG proteins and the POL proteins in this part here of the sequence, and this part over here we'll have the POL genes, so over here. And then down here we're going to have a regulatory sequence called the RRE. And I'm not sure I actually described the RRE in previous videos. It's in the notes, but this is important for messenger RNA transport out of the cytoplasm, no, out of the nucleus and into the cytoplasm, so it can be translated. And not all RNAs need an RRE, but there are two components, at least two components of the lentiviral vector system that require an RRE, and this is one of them. And this will be the poly A addition site for RNA polymerase II. So this plasmid, when it's put into the packaging cell, and it will give rise to, it will be transcribed by RNA polymerase II to make a messenger RNA that has a cap and a poly A tail, and it has a start codon, AUG, and it has a stop codon for the GAG protein. Now remember, if you go back to the HIV-1 video, the pole sequences here, they're only translated if a frame shift happens. So let's say this is going to be the frame shift site. So -1 frame shift, ribosomal frame shift. So that's a little complicated, so make sure you check out the video on HIV-1 biology. So what happens, the ribosome starts translating here, and 95% of the time it's going to stop here. And that's going to give rise to a polyprotein called PR55 GAG, and that polyprotein has the matrix protein, the capsid protein, the nucleocapsid protein, and another one called P6, which is supposed to be important for budding of the viral particles. So 95% of the time this is translated, we're going to get this protein right here, this polyprotein. 5% of the time, when the ribosome is translating this messenger RNA, it's going to slip right here, and it's going to back up one position. That causes the reading frame to shift. By the stop codon is no longer in frame, so it now codes for an amino acid after, if the ribosome shifts. And this is going to be the stop codon down here. And so 5% of the time, and when that happens, we lose this, my understanding is we lose this P6 protein. We still get the matrix protein, we still get the capsid, we still get nucleocapsid, but then we get the protease, we get the reverse transcriptase, and we get the integrase. And these are all connected, and they need to be separated from each other when the virus matures, and they will be separated from each other by this protease right here. This big polyprotein is called GAG pol. So we have a lot of the structural proteins of HIV-1 encoded in the packaging plasmid. And so those packaging cells are going to be making these proteins. So now we have the envelope plasmid. So same thing, circular plasmid. Here's the part that encodes the envelope, the gene for the envelope proteins. Now, there will be a promoter for expressing the envelope proteins in the packaging cell, a +1 site, and then we'll have the coding sequences down here for the envelope proteins. So I'll put an AUG here and a stop codon here, and then we'll have a poly A site right here. Let me back up to the other one. I forgot to say this is where the RRE will be in this messenger RNA, and the RRE helps this messenger RNA be transported to the cytoplasm. This one right here, envelope plasmid, isn't going to need, doesn't have an RRE. So when this is transcribed by RNA polymerase II, we'll get a cap, we'll get the start codon, the stop codon, and the poly A tail. And my understanding is there is no RRE in here, and this just makes it out to the cytoplasm just fine without an RRE, which makes sense. Now, our cells make tons of messenger RNAs, and you know, as far as I know, only HIV messenger RNAs have the RRE signal, the RRE sequence. So, okay, when this is translated, if this is the envelope protein, the envelope gene from HIV1, we'll get a polyprotein with GP120 and GP41, and when this is integrated into the membrane, this will be cleaved by a host protease. It's not important, but the name is furin, so this is a polyprotein that's not actually processed by the HIV1 protease. It's processed by a host protease. Now, if we use a plasmid that has the HIV1 envelope gene, well, we're going to incorporate these proteins into the envelope. And then that virus vector that we're building, it will be great for transducing T cells with the CD4 receptor. However, we can use different plasmids. We can use one that has the VSVG envelope protein, which stands for vesicular stomatitis. How do you spell that? Stomatitis virus G envelope protein. And this envelope protein will help our viral vector fuse fuse with and infect. I guess infect, is that the right word? So transduce a greater, a broader range of cells. So I guess this virus infects a broader range of cells when compared to HIV-1. So if we wanted to transduce a broad range of cells, well, we could use the envelope protein from VSVG. So let's assume we're doing that with our hypothetical lentiviral vector. So we're using an envelope plasmid that encodes the VSV gene. Okay, instead of the HIV GP120, encodes the VCVG protein instead of the HIV1, GP120, and GP41 proteins. So we have 2 more plasmids to go. The third one is the regulatory plasmid, and this one's relatively easy to understand. So it is going to encode the REV gene from HIV1. So promoter +1 site for the messenger RNA, coding sequences for REV, and a poly A addition site. No RRE in this one. I guess this one makes it out to the cytoplasm just fine. So here's the messenger RNA, cap, poly A tail, start codon, stop codon. Now, what does REV do? So REV is the protein that transports messenger RNAs with an RRE sequence from the nucleus to the cytoplasm. So REV binds the RRE site in HIV-1 mRNAs or lentiviral vector mRNAs. And as we have seen so far, the packaging messenger RNA with the GAG and POL proteins that had an RRE. What does RRE stand for? Rev responsive element. And apparently it's really important because we need a regulatory plasmid with the 3rd generation lentiviral vector system, and that regulatory plasmid only encodes the Rev gene. So it seems to be really important. So the 4th plasmid, probably the most complicated plasmid, is the transfer plasmid. Now this is the one that has the information we need for that GFP protein. And so it's going to have our transgene in it, the gene we want to put into our target cells, or the gene we want to put into the cells of a human with a genetic disease, if we're using the lentiviral vector for gene therapy. So let me diagram the different important parts of the transfer plasmid. So there will be a promoter for expression of a transcript in the packaging cell. Let's say this is the +1 site for this promoter. We're going to have a partial 5' LTR from HIV1 up here, where there will be the R sequence followed by the U5 sequence. Now we don't need that primer binding site. No, we do need that. I'm not going to diagram that there though. We still need that, but that's extra details we don't need to talk about right now. So R, U5, and then down here is going to be something I didn't really talk about in the previous videos. So the Psi sequence in an RRE right here for Rev, to recognize the messenger RNA and pull it out from the nucleus, transport it from the nucleus to the cytoplasm. This is the Psi packaging signal. So this is what is used to actually help the messenger RNA be incorporated into the virus particles. So here we're going to have another promoter. We're going to call this promoter B. Let's call this promoter A, and down here promoter B. So there'll be another +1 site right here for this promoter, but this isn't really going to be used in the packaging cell. This is going to be important for the cells that the virus infects, that the lentiviral vector transduces. Downstream of this +1 site, that's where we're going to have our transgene. So the gene we're trying to put into the target cells. In this case, in our example, we're using GFP, but you could theoretically put any gene in there. Then down here, we are going to have the 3' LTR, which is going to have a special sequence called delta U3. So delta U3 is a special U3 that's had some sequences deleted from it, and I'll talk more about that later. So delta U3 leads to a SYN vector, so self-inactivating vector. I'll talk about that towards the end of the video. So delta U3, the U3 is still there, just some parts of the U3 sequence have been deleted. So sort of a mutated U3 sequence. The R region, and then the U5. Make this a little longer here. Now, when this is transcribed from the transfer plasmid that's in the packaging cell, the RNA is going to look like this. Let me erase this here so I can make some room. Whoa. So remember, the poly A site is, in this U5 region right here, just like with HIV1. So U5 isn't actually in this messenger RNA. So we're going to have the R sequence right here, isn't in the 3' end, but it's right here, right? U5 right here. We're going to have the RRE and the Psi packaging signal over here somewhere. we're going to have promoter sequences of promoter B right here. And this is RNA, right? So this isn't going to work as a promoter in the RNA state. But when it is turned into a DNA and put into the genome of the target cell, it will function as a promoter. We're going to have the +1 site for where transcription will start. Once this is integrated into the genome of the target cell, we'll have the GFP coding sequences down here, and we'll have the 3' UTR that's going to have delta U3 followed by R. I'll color this in. So this is the RNA that is packaged into the LVV. Sorry. Okay, so we covered the 4 plasmids. So let's take a quick look at how the vectors are assembling in the packaging cell. So inside the HEK293 packaging cells that have the 4 plasmids, that have the packaging plasmid, that have the envelope plasmid, the regulatory plasmid, and the transfer plasmid. Okay, so what's going on? All these plasmids are having those important regions transcribed and turned into messenger RNAs that are making proteins, in the case of the packaging, envelope, and regulatory plasmids. And the transfer plasmid is making that RNA that's going to be incorporated into the LVV. Still recording? Yes, I am. So now we've seen a lot of this already when we looked at, I think it's lecture 29, video 1. We covered of the HIV-1 virion. So the virions assemble at the plasma membrane, and so the lentiviral vectors will be assembling at the plasma membrane. So I might have to speed up the video here, but what I'm going to diagram first, what I'm going to show is the assembly of the virion with respect to the, what is this one called again? The PR55 GAG protein, and the GAG pol protein. Remember, 95% of the proteins are in this form, 5% are in this form right here. So I'm going to show assembly of this with respect to these, and I'm also going to show the, what did we decide? We decided the envelope protein we're using is the VSVG envelope protein, which is going to allow our lentiviral vector to fuse with lots of different target cells. So let me diagram these. Okay, so here we're looking at assembly of the lentiviral vector in the packaging cell. We have 95% of these proteins here are the PR55 GAG polyprotein, and 5% right here, I'm just showing 2, are the GAG pol polyprotein, and the GAG pol polyprotein is the one that interacts with the transfer RNA, so the RNA that holds the GFP transgene and the LTRs and things like that. And so you can show you 2 different RNA, or 2 identical, I should say, messenger RNAs are being incorporated into the lentiviral vector. And we have the VSVG envelope protein, which has been incorporated into the plasma membrane of the HEK293 cells, the packaging cells, and because of the budding process, we're going to end up with these VSVG proteins in the envelope of the lentiviral vector. So remember, the envelope is derived from the cell membrane of the packaging cell. So here's the protease, right? And the protease doesn't become active until this buds from the packaging cell. And after that happens, and that protease gets to work, we end up with something like this. So this is our mature lentiviral vector, which has the GFP transgene that we wish to put into a target cell that is receptive to fusion of our LVV, I should say receptive to fusion with LVVs that carry the VSVG envelope protein. I changed the shape of the VSVG protein in this diagram relative to the last one. But we can see, okay, let's take a look. This looks a lot like the HIV-1 virion, right? So it has an envelope protein. It has the envelope derived from the host cell, the packaging cell in this case. It has the matrix protein. It has the capsid, which holds the genetic material. The capsid proteins are in blue. Inside the capsid, we have the nucleocapsid interacting with the RNAs. Only one of the RNA molecules is diagrammed here. We have the integrase proteins. Just one is shown. The reverse transcriptases are there. Just one is shown. And the proteases are inside here. Just one is shown here. So what's different? Well, this thing right here. This RNA normally has, right, the HIV-1 genomic RNA, so the genetic material that makes HIV-1 infectious. But there's no HIV-1. This is not the HIV-1 genomic RNA. It has little bits of the sequence in there, right? Like partial LTRs are in here. We have a couple other things. We have the Psi packaging signal. We have the RRE. But really, there's not that much of the HIV-1 sequences in here. What's in there is the sequence of the GFP transgene and the promoter that we need for the target cell to express that GFP transgene. So when this lentiviral vector is used to transduce a target cell, that target cell isn't going to make more of these particles, right? Because it doesn't have the genetic material to make any of these proteins right here. It's just going to get the genetic material for expressing GFP. We can say that again. So remember, there were 4 plasmids, right, for making these. So the 4 plasmids were the packaging plasmid, which made the GAG polyprotein and the GAG pol polyprotein. So none of that information is in here. So the regulatory plasmid, which had the information for making REV, for transporting the RNA here out of the nucleus and into the cytoplasm. Well, the sequences necessary for REV aren't in here. How about the envelope plasmid? The sequences necessary for making the VSVG protein. Those sequences aren't in here. So only sequences from the transfer plasmid are inside the LVV. So we don't need to worry about the target plasmids making more of these. The target cells that are transduced by this lentiviral vector being able to make more of these particles. So they're going to get the transgene, the GFP transgene, and that's about it. So one other thing I would like to cover This video is kind of long. I'm sorry about that. This is a really complicated subject, and I hope you're understanding how it works. So now what has to happen? So essentially, we can take these purified lentiviral vectors and mix them with the target cell, and essentially this RNA is going to be turned into a DNA by the reverse transcriptase, using the same similar mechanism that HIV-1 uses to turn its RNA into a DNA. So similar mechanism, and then it's going to be transported to the nucleus of the target cell. This capsid is going to go through the nuclear pore. Capsid is going to dissolve, and then that DNA, after this RNA is turned into a DNA, that DNA is going to be incorporated into the genome by the integrase, using the same mechanism that is used by HIV-1 to integrate its DNA into the genome. So very similar mechanisms going to occur, and what we're going to end up with is the transgene in the genome of the target cell. And what is it going to look like? So we're still going to end up with that 5 base pair target site duplication, because the integrase, that's a feature of the HIV-1 integrase. So what else is going to integrate here? We're going to get the 5' LTR and 3' LTR. Now remember, if you go back and watch how HIV-1 reverse transcription mechanism restores the full-length LTRs. So let me just put some stuff in here. This right here would be the delta U3. Here will be R, here will be U5, here will be delta U3. So if you remember, the RNA particle inside the virus only had what? Only had an R, a U5, and a delta U3 over here, and an R. So that was the RNA. Now the reverse transcription process restores the full-length LTRs. But because this is delta U3, we're going to see a delta U3 here and delta U3 here. So make sure you watch the video on HIV-1 reverse transcription to understand how these LTRs are regenerated. So the lentiviral vector is going to regenerate the LTRs. This is going to be inserted into the genome of the target cell. There will be a promoter here, and then there will be the GFP sequences down here, or whatever sequences of the transgene that we wanted to insert into the target cell. And that's essentially it. So this is, I wanted to diagram this because I want to talk about the self-activating nature of these lentiviral vectors. So this delta U3, so it's only a partial UC3 sequence, and HIV-1 uses the U3 sequence. It uses a promoter sequence in there. So if this was HIV-1, this U3 right here in the 5' LTR would have the promoter needed to transcribe this whole thing and make the HIV-1 genomic RNA that is packaged into the virus. So researchers and scientists have figured out that if we use this delta U3 sequence in the lentiviral vector, then there's no promoter sequence here. So the target cell cannot make, or should not make, a long messenger RNA that includes the LTR sequences at the beginning and the end of the RNA. And the reason they wanted to do that is they don't want anything, they don't want any RNAs that look like a lentiviral RNA. Lentiviral RNAs have LTR sequences at the beginning and the end. They didn't want that. They wanted to reduce the chance that this could look anything like a lentiviral RNA, so they used these self-inactivating vectors. The delta U3 sequence does not have a promoter, so the only RNA that this should be able to make in the target cell is an RNA that's transcribed using this internal promoter here, promoter B, I called it earlier in the video. So the only messenger RNA this should make should look like this: 5' cap, GFP, or whatever gene we wanted the target cell to gain by transduction with the lentiviral vector. and then it will then here, let's say this is the stop codon, this will be the 3' untranslated sequence, and there'll be some delta U3 sequences in here, and some of the R sequences and the polyase termination signals inside the U5. Okay, so those cells will have this they'll be making this messenger RNA here, and it'll be exported to the cytoplasm. Ribosomes will translate it, and the cells will be glowing green. Okay, so that's essentially how the 3rd generation lentiviral vector system works. Now, if you understand that, you're going to understand this new gene therapy approach that I'm going to talk about in the next video, and it's a gene therapy called CAR T therapy, and it's used to treat a form of leukemia. Okay, see you in the next video.